Okay, so in this video we're going to talk about neurulation, vesiculation, basically embryology of the brain and the spinal cord on how it develops. Okay, so if you remember, we talked about this a while ago, gastrulation. Gastrulation occurs in about the third week of development of the fetus. And what is gastrulation? So all it really was if you remember, we have a fold right here, right? We have this bilaminar streak. This is around week 2, the week of 2s. And then around week 3, we have another line, right? We make a trilaminar disc. So this trilaminar disc came from embryology development and stuff like that. But what was important about this trilaminar disc in week 3? Well, we had a neural crest cell, right? We had a neural, we formed a neural line called the primitive streak. This primitive streak, right, caused like an invagination. So what is that? So like the structure kind of like folded inwards. Kind of looked like this. I don't know, I draw like a bird, that's how I remember. Like that. So these 3 lines folded inwards on each other, this little invagination. Now, this is around week 3. If we continue proceeding down for the development of the brain and the spinal cord, it's all coming from this. It's all coming from the same exact process. So now, before we talk about it, this structure right here, I just want you guys to remember, the top line is the ectoderm. So the top line is the ectoderm. The middle line is the mesoderm. See how I have a dash right there? Because the middle of the mesoderm is going to be the notochord, a very important structure. So the middle line is the mesoderm. The green circle is the notochord. And then on the bottom, you have the endoderm. Okay, makes sense. Ecto for the outermost, meso for middle, endo for innermost. Not bad. Okay, what else is important to talk about? Where does this neural, where does the groove, the primitive streak, take place and how? Okay, so it's the formation of the 3 germ layers, known as gastrulation, and the primitive streak occurs on the epiblast. So here's our epiblast, the 3 lines, primitive streak forms. Then begins neurulation. What is neurulation? Neurulation is going to be forming our whole entire brainstem and our spinal cord, the structure. Okay, how does this occur? Well, it happens around the 4th week of development. So the early 4th week, or around day 22, 23. Okay, now, if we proceed down, we see something called ectodermal proliferation. What is ectodermal proliferation? Okay, so the ectoderm is going to proliferate into different things. Not bad. What is the ectoderm going to proliferate into? Well, part of it's going to be parts of the brainstem. The other part of it's going to be parts of the spinal cord. What about the mesoderm, endoderm, and the notochord? Well, it's also going to deviate into different things, which we're going to talk about. All right, but to continue first, ectodermal proliferation uses homeobox genes. So all these genes are, well, where are they located? They're located in the notochord, precordial plate, and the neural plate. And basically they tell the epiblast, or the trilaminar disc, these 3 lines, how to fold, how to orient themselves, and how to shape themselves. Okay, and they're going to form the surface ectoderm, like we said, the paraxial mesoderm, middle, and the notochord, that circle right here. Okay, where is that? Okay, so now, boom. Okay, so how does this neural plate form? What happens? So say it's a straight line, right? You have 3 straight lines. The ectoderm layer, the top part, where the neural plate is going to form or function, it's going to be like this, right? So now from underneath, right where the notochord is and stuff, it's going to be pulled. So it'll be pulled down, and it'll fold like this. Okay, so it's straight, and then from underneath where the mesoderm is, it gets pulled down. What happens when it gets pulled down? Okay, if it gets pulled down, the neural plate is going to start forming neural folds. A fold, right? It's folded upward, it's like a round structure. It's a fold. Okay, so this fold then is going to have neural crest cells right near it, right to the right and left of it, right around this neural fold is going to be neural crest cells. So right in this paper. Where are they going to be derived from? Well, they're going to come from the neuroectoderm. So the neural crest cells are going to be right near the fold, and it's going to be known as the neuroectoderm. These neural crest cells are going to deviate into a lot of different things, which we're going to talk about. But let me just continue this process first, and then I'll explain. What else? These neural folds, see I have the orange right there? And the crest, these neural folds are going to form like a little, like, I don't know, divots kind of here, like a bridge. And this bridge eventually is going to collapse. When the bridge collapses, this whole entire neural plate, which went from here to a neural fold to the bridge, is going to form the neural tube. So if you proceed down, you can see top part, right? You have the ectoderm. Middle part. Okay, what is this middle part? Well, this is where the neural plate got sucked in. When it got sucked in, had the neural folds with the neural crest cells, forms the neural tube. It folded, the bridge broke, and now you have the circle. Or the tube, whatever, however you want to think of it. This neural tube is now formed. But when this neural tube is formed, does anything happen to the neural crest cells? No. They just stay on the perimeter. They stay on the outside of the neural tube. Okay, so let's look at the structure. You have ectoderm, neural crest cells, neural tube. What's below him? Notochord. You're not getting rid of the notochord. He's still got to stay there. Notochord. What's underneath the notochord? Endoderm. Okay, what's to the left and to the right of this neural crest cell? Or to the neural tube? The mesoderm. You still have the mesoderm. Like, you can't just like get things away, kick things out. This is not like the pharyngeal apparatus where the fifth arch leaves. That does not happen here. Everything stays. Okay, so now the neural folds, right? The orange ones, they fuse together. So remember I said the neural folds are like a bridge that like holds this thing apart, like not apart, but like say it's like that, it holds it together. Well, when it goes away, doesn't disappear, what happens? They fuse together and they stay in the neural tube. So this bridge is inserted like this now. I don't know if it's the best way to look at it, but this is how I kind of like process it and remember it. Okay, so this view, where this occurs around day 22 to 23. All right. Basically the same day as the start of neurulation. Now, this is the view what it looks like laterally. Now, if you're looking from like, say above, you know how I like looking at things from the above angle? This is what it's gonna look like. I know this really doesn't look too good and it's not drawn too neatly, but just like try to understand and process what I'm trying to say here. So the middle part of it's gonna be that neural tube. Remember we drew the tube like this? It's right here. Well, if you angle it this way, the middle part is the neural tube. So if you're looking straight down, you can see the neural tube. Makes sense. And then on the top part of it, like this blue area, it's going to be more exterior, like outside of it. That's going to be the ectoderm. Makes sense. Outside ectoderm. And then I drew the endoderm like around this whole entire sheet because it folds up. It like folds around it. I don't know. Couldn't really draw it too artistically, but I tried. What else is going to be around the neural tube? Neural crest cells. Makes a lot of sense. Okay, so what's important now about this neural tube and the neural crest cells? Well, if you see the structure looking like this, right? Kind of like a straight up, like a, I don't even know, like a tube, like a pipe, I guess, running up and down. You're going to have a cranial end and a caudal end. What's going to be the cranial head? Well, it's going to be top of my body, and the caudal is going to be the bottom, like where my butt is. Because that's all we're forming right now. We're not forming the legs and the arms. We're just forming the bottom of the spinal cord to the top of the skull. So cranial and caudal. Okay. So if you see the cranial end, you know it's the caudal end. That's how you orient yourself. Boom. You see the neural tube. You're like, okay, what is the neural tube going to form? He's going to form this central nervous system. What's the central nervous system? Brain and spinal cord. All right. So now you have the brain and the spinal cord. But what else comes from the spinal cord? You're missing some stuff. Or you could say I'm missing some stuff, right? Well, if you keep going down, this looks like this tube, right? Well, what's around this tube? The neural crest cells. What are the neural crest cells? Well, they're going to make the peripheral nervous system. PNS and ANS, parasympathetic and autonomic nervous system. Makes sense. Neural tube, right? Brain and spinal cord, the middle. On the outside of him, right here, remember this picture on the outside, the borders? Well, right here on the borders, peripheral, away from the spinal cord, the nerves that are flowing down to my fingers, down to my foot, etc. It would make sense for it to be on the outside. That's going to be the neural crest cells. Okay, so the neural tube, central nervous system, brain and spinal cord. Neural crest cells, peripheral nervous system, and autonomic nervous system, peripheral nerves. Boom. Now, one more structure I want to talk about. I'm going to go into a little more detail, but I just want to mention it real quick. This neural tube, right? Say I took him out. I took this guy and I put him over here. Okay. fine. Where am I going with this? You have a cranial end, you have a caudal end, top and bottom, top and bottom. So now this cranial end, right, is called the cranial neuropore. Neuropore, what does that mean? Well, if it's neuro, it should be something with this neural tube, and the pore is just like where something inserts and exerts out of. This cranial neuropore is supposed to close around day 25. That would be a healthy baby. Around day 25, this neuropore is going to close. Now, on the bottom, you have the caudal neuropore. Good. It's just the bottom part, right? Right down here, bottom. Okay? The caudal neuropore is supposed to close around day 27 to day 28. Now, what happens, or like what makes them close? So, the importance of these neuropores is just at the closure of the nervous system. How does it close? So, when a mother is pregnant, she needs to take folate. Like, this is a supplement that she needs to supplement to her baby. Just proper vitamins. Folate closes these neuropores. Boom, not bad. Now, if she does not have folate, right, and and you do a test, and the test result shows a lot of acetylcholinesterase or alpha-fetoprotein, what's going to occur? Well, that means there's something wrong with the fetus. Something is wrong with development. High levels of alpha-fetoprotein or high levels of acetylcholinesterase means there's something wrong. Now, if you have a problem with the cranial neuropore, so like the top part doesn't close, it's very, very serious. You're going to develop anencephaly. What is anencephaly? Well, basically the forebrain and like the brain doesn't close. So the brain is exposed to the air, the forehead doesn't develop, the forebrain doesn't develop, the baby can't survive. Like, you can't live through that. Very, very severe. Now, what happens if the caudal neuropore does not close? Well, if the caudal neuropore doesn't close, or doesn't properly close, however, sometimes it just partial closure, sometimes non-partial. If it's like a partial closure, you're going to develop spina bifida oculata. What is spina bifida oculata? Okay, well, this is the neural tube, right? It's another neural tube defect. You're going to see acetylcholinesterase and alpha-fetoprotein, and this is going to cause tufts of hair or a dimple on the lower back. So, like, if you see like a baby, like we're in a picture, or somebody has like a little tuft of hair or a dimple right in this region, they have spina bifida oculata. It wasn't a proper closure of the caudal neuropore. Boom. Now you also have meningocele, you have myelinomeningocele, and myeloschisis. What are these differences? Okay, meningoceles, or just if you have a meningocele defect, this is a meninges defect. Just think, meningocele, meninges, boom. What happens? Okay, so the meninges are herniating out of the back. Okay, but just because they're herniating out doesn't mean they're connected to the outside air. They're just connected just like a little herniation, right? A little bubble. Now, if you have myelomeningocele, what does this happen? What happens here? Same reason, this caudal neuropore doesn't properly close. However, now you have the spine and the meninges herniating out. Okay, meningocele, just the meninges. Myelomeningocele, meninges and spinal cord herniating out. Okay. Spina bifida oculata, dimple or a hair tuft, right? The least severe. If you see, I'm going from least severe to most severe. So spina bifida, least severe. Meningocele, just meninges. Myelomeningocele, meninges and spinal cord. Most severe now, myeloschisis. Okay, why is this the worst one? Well, it kind of sounds pretty bad, right? Myeloschisis, whatever. But this is the worst one because there's a full exposure to the outside environment. What happens? The meninges in the spinal cord are herniating out, right? But instead of skin or meninges like covering the spinal cord, like in the first ones, the meningocele and myelomeningocele, this time they have a full exposure to the outside environment. So there's no skin or no meningeal covering. Okay, so the outside environment is exposed to the person's spinal cord. Very, very dangerous. And now, which I already mentioned, I just want to say descending order: spina bifida oculata, meningocele, myelomeningocele, myeloschisis, most dangerous, anencephaly. Anencephaly is the only one who's going to be in the cranial neuropore. The other ones are going to be in the caudal neuropore. Boom. I'm going to talk a little bit more detail about these things at the end of the video, but I just wanted to mention it real quick right now. Okay, so I want to just cover 3 questions based on what we just went over. So in the late 1990s, in an attempt to reduce the incidence of a specific category of birth defects, the U.S. Food and Drug Administration mandated the supplementation of grain products with a coenzyme essential for nucleotide synthesis. This coenzyme is absorbed in the jejunum. The chemical exerts its greatest effect on which of the following regions of the developing embryo? Okay, so we know it's something of birth defects, and then we know it's a supplement, and we know it's a coenzyme. Okay, it's used for nucleotide synthesis, whatever. All of this stuff we know. That's what they told us. But what is this really asking? So if it's a birth defect, what did we say the birth defects were? Well, there were spina bifida, anencephaly, the myogenocele, meningocele, etc. So what didn't close? So it was the caudal, or that's the cranial and the caudal end didn't close. Okay, what causes these ends? Well, folic acid. Okay, so the supplement they're probably talking about is folic acid. So the chemical effects exerts its greatest effect on which of the following regions of the developing embryo? Well, we know it's this region, right? And what region was this? Well, this looks like the neural tube, because that's what needs to close. So if any of the answers neural tube? No. So now we have to go process of elimination. Where does the neural tube come from? Okay, well, if we remember the drawing, let me draw it up here. We had the 3 lines, right? That 3 lines then went like this. That's really bad. The trilaminar disc, I'm sorry. Let me try to redraw it real quick. Okay, and then when they separated, right, it was like this, the center. Okay, so you have the ectoderm, the meso, and the endo. But what was in the middle? All right, the notochord was in the middle. Okay, and then the process continued down, and what formed? Well, the top region, right, we said this top region gets pulled down, the piece of paper example I gave. And this top region formed the neural folds. The neural folds then formed what? The neural folds then formed the neural tube. And the neural tube looked like that. So this region right here is the neural folds, right? Well, the beginning process of the neural folds. That piece of paper example I gave. Okay, so the only place that folic acid would take place is going to be where? The neural tube. And the neural tube comes from the neural folds. So that's the answer. So notochord's wrong. Midgut's wrong, med and froze are wrong, and meso and phrenic tubules are wrong. The only reason notochord you would think might be right, because you know it's from the mesoderm and you remember that circle, but you have to remember that that circle, this notochord, does not form the neural tube. The neural folds is what formed the neural tube. So let's look at our answer. That's right, yeah. Okay, folic acid was the thing, the coenzyme, and yeah, spina bifida, anencephaly, neural tube defects, we said that. Here's a better picture. So if we want to look at it real quick, you have the neural plate border. This is the primitive streak, goes up here. This folds down like the piece of paper I said. The neural folds form together, right? When they go together, what happens? They converge, like it says. And the neural groove, right? It connects to the neural folds, and this is going to form the neural tube. So this is going to be the neural tube. And this is like the side angle. So if we took it like this, it would look like that. And then you would have the caudal. So say this is the caudal part. And then if you came to the back side back here, could be the top. This would be the cranial part. Okay, so that's not too bad. And what are these green dots around it? Well, these are the neural crest cells that we spoke about before. Okay, let's see another question real quick. A research team is studying gastrulation in mammalian embryos that have similar development to human embryos. Good. The team plans on assessing the migration and differentiation of epiblasts into the ectoderm. Okay, so epiblasts into the ectoderm. So we know it's epi, and then we have to go to the ecto. Okay. Mesoderm and endo. Okay, so we're going into all of them. What are we going into? The trilaminar disc. Remember the epiblast forms the ecto, meso, and endoderm? Okay. And then I should have kept reading, but it says the trilaminar embryonic disc. This will be done by tagging the epiblast in a specific region of the embryo. Okay, so we're going to tag this guy. We're going to tag him. The tagged epiblast will most likely migrate through which of the following embryonic structures? Okay. Let's see. So if we have this trilaminar disc and then it forms into the notochord in the middle, right? That fold, then it processes down into what? Well, it formed the neural tube. The neural tube had the ectoderm around it and the mesoderm around it, right? Let's see. The cloacal membrane. I don't really know where this goes, so maybe. The neural tube, well, definitely that's an option. The precordial plate, maybe. I don't think so, though. Precordial plate has nothing to do with that. The primitive streak, well, yeah. The primitive streak starts the epiblast. But the primitive streak, I don't think that goes into another structure, but let's keep looking. And the yolk sac, no. The yolk sac comes from the notochord, so that's wrong. Okay, so we have the ectoderm, mesoderm, endoderm. Keep going down, and we're forming what? We're forming the neural tube. The primitive streak is what starts the epiblast forming, so that's not right. So I'm pretty sure the answer is neural tube. Never mind. Okay, so we have the ecto, meso, and endoderm. It says the tagged epiblast will most likely migrate through which of the following embryonic structures? Well, let's remember, what's the first step? We have the trilaminar disc, right? The epiblast gets tagged, so the epiblast makes these three things. How? So when the epiblast, say, looks like this guy, right? Strong thing, epiblast, right? Primitive streak comes up the top on top of it. When the primitive streak forms, okay, it separates into three different layers, that trilaminar disc. And it makes the ecto, meso, and endoderm. So I believe the tagged epiblast will most likely migrate and form the primitive streak. The yolk sac, this is going to come from the notochord, so this is wrong. The precordial plate, no. Cloacal membrane, no. And what about the neural tube? Well, the neural tube does process from this this trilaminar disc region, and it is going to form the neural tube after the neural folds form. Right? Remember the neural folds we spoke about right up here? So after the neural folds form, and we're going to form the neural tube, this is after the primitive streak, the first step. But I believe the first step is the primitive streak, not the neural tube. The neural tube is not the first step in this process. So primitive streak is going to be our answer. So here we go. The primitive streak is a linear accumulation of epiblasts. Yeah. So the epiblast is like this. 1, 2, 3. We'll connect it together. Primitive streak forms, separates into three different layers. Trilaminar disc. Good. Okay. Last question. Now, if you do not know the histology or like how you could see these pictures, you don't need to answer this question. So a research study examines brain tissue from an autopsy specimens of patients with HIV and AIDS. The circles in the slide show that the cells are activated in response to the CNS or central nervous system damage. So this is all we need to know. Something with the CNS. Boom. Which of the following embryonic germ layers is the most likely origin of these cells? Okay. So CNS, germ layer, answer. So if we remember, we formed neural tube, right? Around that was the neural crest. Those are those neural crest cells. What was around that? Well, we had the mesoderm, we had the ectoderm, etc., right? Okay, but this thing didn't really form stuff with the central nervous system. This didn't form central nervous system. What did form stuff with the nervous system? Okay, we knew the neural... Oh, that's a bad color. It's too big. Okay, we know the neural tube formed something with the nervous system. And we also know the neural crest formed something with the nervous system. But what? Well, if you forgot, just think of it, just look at the picture. The middle part looks like the middle, obviously, and the middle of your spinal cord or your nervous system is the spinal cord, brain, and spinal cord is the middle. On the outer side, what looks like on the outside? Well, that'll be peripheral nerves going to your limbs, your arms, your hands, etc., right? Legs. Okay, so if you know that the neural tube is the middle, spinal cord and brain, and the neural crest is peripheral, like the limbs and stuff. Well, peripheral is PNS and ANS, parasympathetic and sympathetic and autonomic nervous system. Neural tube for spinal cord and brain, you know this is the central nervous system because just have to memorize central nervous system is the brain and spinal cord. Okay, so let's look through the answer choices. Which of these germ layers is most likely the origin of these cells? Endoderm? No. Mesoderm? Maybe. Neural crest? Well, you know it's not neural crest because we're talking about central nervous system, so that's wrong. Neuroectoderm? Maybe. Surface ectoderm? Definitely not. Okay, so now we have to decide between neuroectoderm and mesoderm. How are we going to decide that? Well, what did I say about the neuroectoderm? What does he form? Well, the neuroectoderm forms the neural crest cells. And what does the mesoderm form? Well, the mesoderm is going to pull what? The piece of paper example like we talked about before. It's going to pull from the top region and it's going to form the neural folds, which are then going to form the what? The neural grooves, and they're going to form together to form the neural tube. Okay, so now we have neural tube and we have neural crest cells. Which one's which? Well, we said neural tube is CNS, brain and spinal cord. And we said neural crest is what? PNS and ANS. So neuroectoderm can't be the answer. What is our answer going to be? It's going to be the mesoderm. Let's see if that's correct. Yes, that is correct. The mesoderm. Okay, how is this the mesoderm? Well, we just talked about how it goes, how the whole entire flow happens. So if you don't have this memorized over here, don't worry about it. Just break it down like how we talked about it. I lied. I think we have another question. Okay, one more question. A 19-year-old woman delivers a male baby at term. On examination, the child is noted to have a cystic swelling over the lower back and the midline, but appears to have both motor and sensory modalities intact in both the lower extremities. Okay, cystic swelling in the back. We have to automatically but we have to think, okay, spina bifida, meningomyocele, right? The meningomyocele. It could be encephaly or myeloschisis, right? One of those four things we spoke about. Okay, so let's keep reading. Just keep this in the back of our mind. Okay, over the lower back and the midline, but appears to have both motor and sensory modalities intact in both lower extremities. MRI scan reveals a cystic swelling in the lumbar region with no evidence of neuronal tissue in it. Okay, so no neuronal tissue. What does that mean? Okay, so if there's no neuronal tissue, that means it can't be the myelinogenocele, right? Because he was spinal cord, and what else? And meninges. Okay, so if it can't be one of those, we have to keep going. The child is diagnosed with meningocele. Okay, so they told us we should have kept reading, but with a meningocele, a failure of which of the following has resulted in this defect? Okay, so we have to remember 4 of them. What 4 come from the caudal, and which one comes from the cranial? Okay, so, I'm sorry, I said that reverse. What 4? Yeah, no. What 4 comes from the caudal? So we're going to write, over here, we're going to write caudal. And caudal was spina bifida. Boom. What else? Well, it says right here, meningocele, right? What else? Myelinogenous. I can't pronounce or spell. Bear with me. Okay, and what was the 4th one? Myeloschisis, right? Okay, and then which one comes from the cranial side? This is a little extra. You don't need to know for the question stem, but I figured it would help. So the one that comes from the cranial was encephaly. Boom. Okay, so now it says we have meningocele. Meningocele is which one? Right here. Where did this occur from? Well, we know it's from the caudal end. Okay, so we could say we have to find caudal. So this is cranial, so that's wrong. Caudal, so probably. Disappearance of notochord, no. Formation of neural tube, well, maybe, because this is where these processes occur from. Separation of the prosencephalon, no. This is not even what they're asking. So it's between B and D, right? Now, what are birth defects from? Well, birth defects are from failure of the neural tube to close. So yeah, it's formation of the neural tube, but the question is asking for a birth defect, and that is from the closure of the neural tube. And you have a caudal neural pore, which is on the bottom, and the cranial neural pore on top. But it's asking for a meningocele, and meningocele is caudal. So our answer's got to be B. Let's see if we're correct, and yes, we are. Okay. Now for the fun stuff, or interesting stuff. I at least think it's a lot better than what I just spoke about. So we have to remember everything I just made, right, or everything we just talked about, that little structure from the neural crest, the neural tube, etc., right? So he's going to form now. That neural tube is going to differentiate into our brain and spinal cord. How is he going to do this? Okay, so remember I had the cranial end, the caudal end, right? And the little, the neural tube was in the middle of it. Remember let's see, that piece of paper, we had the neural tube, and around it was the ectoderm, the mesoderm, etc. Okay, so we're taking this neural tube now, the little piece of paper that we formed, and we're taking them and we're putting them right here. Boom. Neural tube. Neural tube is now going to differentiate, and he's going to form different things. So the first step, or what he's going to start to look like, is this structure right here. Looks really weird, I know, but this is called vesiculation. Okay, what is vesiculation? Vesiculation is just taking the neural tube, starting to develop, starting to differentiate, and we're just changing into different things. We're going to go into different regions of the body. What parts? So the top part's going to be the proencephalon. The proencephalon is going to form our forebrain. Our forebrain is going to be like the top region, our lobes, etc. The middle part is going to form the mesencephalon. This is going to be our midbrain. We're going to talk about where the midbrain is, but the midbrain is like literally the middle of our brain, kind of about the brainstem. Okay. The bottom part is going to be the rhombencephalon. This is going to form the hindbrain. We're going to talk about him too. Underneath that is going to be the spinal cord. So if you can kind of see with the colors, we have navy blue, royal blue for the proencephalon, purple for the mesencephalon, green for the rhombencephalon, and like maroon for the spinal cord. These guys are going to flow, like if you see the board, I'm going to make the board a little bigger in a little bit, but if you see the board going this way, they're all going to coexist with each other. They're all going to be in the same stage. It's color coded. Okay, so first, I'm just going to start off with the top three regions, right? Literally the brainstem, the brain. The bottom is just going to be the spinal cord, so we're just going to talk about the brain first. Okay, he's not too bad. Proencephalon, right? He's going to then differentiate into two different regions. I know it sucks, but he's going to differentiate into the telencephalon and the diencephalon. Okay. The mesencephalon, thankfully, he's going to stay the same. He's going to be the mesencephalon as well. The rhombencephalon is going to differentiate into two things too. The metencephalon and the myelencephalon. Okay, I know this is not related, but I don't know, for some reason this reminds me of the brachial plexus. If we remember the brachial plexus, we differentiate into two different regions, one region and two regions. Remember C5, C6, C7, C8, and C1. I don't know why, but I just remember mesencephalon for C7 just staying the same. Does not correlate at all, just it helps me remember. Okay, so the prosencephalon divides into two regions, right? Telencephalon, diencephalon. Mesencephalon, just the mesencephalon, stays the same. Rhombencephalon goes into the metencephalon and the myelencephalon. Now, one other thing I forgot to mention real quick. Remember we had the neural tube. The little neural tube that I have the paper structure of. Well, what was around the neural tube? The neural crest cells. Okay, what were the neural crest cells used for? The neural crest cells are for the peripheral nervous system, autonomic and parasympathetic. Okay? The neural crest cells, since that's what they're dividing into, the peripheral nerves, they divide into a good amount of things. I'm going to list them real quick. It's something we kind of just have to memorize, can't really talk through. But they mainly differentiate into the Schwann cells, parasympathetic ganglia of the GI tract. We have the adrenal medulla. We have the meninges. What meninges though? We just have the pia and the arachnoid meninges. Why? Well, if we remember or if you watch the video on the skull and the cranial, the sinuses and the ventricles, cerebral spinal fluid, the dura mater So the dura mater is the matter that has the triangle in it, the venous sinus. He is derived from the mesoderm. Okay, just remember that. But the neural crest cells is going to form the meninges of the pia and the arachnoid matter in particular. Okay, not bad. We also have melanocytes, which is for our skin. We have odontoblasts, which is going to be our teeth. We're going to form endocardial cushions. Now, two clinical things. If you have an improper development of these neural crest cells, like they don't form properly, we could have Treacher-Collins syndrome or Robin sequence. It's another type of disorder. Also, you could have Hirschberg disease, another disorder that comes from underdevelopment or malformation of the neural crest cells. Not too shabby. Okay, I'm now going to keep proceeding down. Don't worry about this bottom structure right now. I'm going to get to them. Actually, we're going to talk about them right now. Spinal cord, right? You have the prosencephalon, mesencephalon, rhombencephalon form this region. Spinal cord, right, is going to go through, it's going to form a primary vesicle is what they call it. It's going to go through the central canal. This central canal is then going to be the neural tube. Wait a second. I just said the neural tube was up here, right? Well, we have to remember the neural tube is from the cranial all the way down to the caudal end. What does this mean? Cranial. Where is cranial? Up here. Where is caudal? All the way on the bottom. Not my feet though, like by the bottom of the spinal cord. Okay, so this neural tube is the whole entire region. All right, so the bottom half of it is going to be the spinal cord. Okay, I know it's kind of like, it's not really directly in half if you think of your body. It would mainly be like this, if you look at it. Like the small region is going to be here, and then the rest is going to be the spinal cord. But for a video and like preference, like just easier to say, I split it in half. Okay, so the spinal cord, and then you're going to have the neural tube. Now, you see these structures, they kind of just like formed into like a bigger thing with like a different shape. Spinal cord, not the same. For the spinal cord, think of we have this spinal cord, right? And we cut it, we just cut it, right? When we cut it, we take the small piece of the spinal cord and we're looking down into it. So here's the neural tube, here's the spinal cord. I take it, but instead of looking at it in this view from the side, I'm looking into it. So I'm looking straight down below. That's what this picture is going to be. Okay. What does this even mean? Well, this neural tube is going to have 3 different layers in it. What? So this is what it is. If here's the structure, right? Here's that like little tube that you're looking straight down, the little piece of paper diagram. If you're looking straight down, you're going to have something in the middle. What's going to be in the middle of it? Yeah, you're going to have bones, the spinal cord, etc. But that's not what we're talking about right now. What's going to be right in the middle of it is going to be these 3 structures. So say these 3 structures, right, I have blown up. If you draw a line, if I draw a line like that, these 3 structures fit into this little circle. Very tight, very squished together, but you can't really see it if you look at it small. You have to see it blown up. So just take the middle and blow it up. Not bad. 3 different layers. Okay, so you have an outermost layer, middle layer, and inner layer. Makes sense, it's pretty self-explanatory. But what are these layers? Okay, so the outside layer, the outermost layer, right? He's going to be called the marginal zone. Okay, what does this mean? The marginal zone is going to have neurons and axons, right? And since he's the outermost layer, he's going to form white matter. He's going to form cell bodies, the spinal cord, spinal ganglia, and brain. What is white matter? Well, white matter is in the spinal cord, remember? It's fast, myelinated, etc. That's going to be our white matter. Boom. Now, the middle layer is going to come from our neuroblast. So the neuroblast, a different type of cell, he's going to form the middle layer. This is known as the mantle layer. Okay. What is this mantle layer going to do? It's called the intermediate zone, and he's going to form gray matter. Okay, just remember that. I'm going to talk a little more detailed in a second, but just remember that. Now, the middle layer, or innermost layer, is the neuroepithelial, right? This is known as the ventricular zone. What is he going to form? He's going to be the ependymal layer, and he's going to form neurons and macroglia. Okay, these layers, right? Now we're going to converge. Not converge, but we're going to deviate even more because we're further developing in our body. We're not just going to stay like this. We have to deviate into different structures and talk about it. Okay, so now if we continue, we have the three structures, right? Deviating into something else. What is it going to deviate into? All right. Looks a little weird, but it's really not too bad if you just bear with me real quick. I know this is my favorite thing, but neural tube, right? This little paper structure. This whole thing, these three layers, right? Condensed, or they're all condensed in this neural tube. Decent. Neural tube right here. Okay, it's in the middle. We see I have the neural tube, right? And then surrounds the neural tube. I have a red region, a purple region, etc. Not bad. The red region is going to be the top, right? This is going to be known as the alar plate. Alar plate is going to be on the top. The purple region is going to be known as the basal plate. The basal plate is going to be on the bottom. What does this mean? I got it. The middle region that differentiates all of them is going to be the sulcus limitans, right? All he's going to do is separate them. Okay. What is alar plate and what is basal plate? Well, if we remember the spinal cord, we have a posterior and an anterior side. Okay. Posterior is known as dorsal. Anterior, ventral, right? So posterior, dorsal. So alar plate is going to be posterior. Basal plate is going to be anterior. Alar posterior, basal anterior. Good. Now, what is this? It further develops into this region right here. This is known as the ganglia, right? Okay, so we have what? This is the spinal cord, a cross-sectional view again. A further developed one though. This top region, right, this red region, is going to develop into the posterior gray horn. Where is she even coming from? Well, these alar plates are converging together and they're forming the posterior region known as the posterior gray horn. Okay. The bottom region, you see it's purple. He's taken the basal plates coming together. They're forming the anterior horn. Anterior gray horn, I'm sorry. Now, what the hell are these things? Well, if we remember, posterior, right? The posterior gray horn, sensory neurons flow into it. Not bad. The anterior gray horn, the motor neurons flow out of it. Perfect. So now we have the neurons coming posteriorly, and then we have the neurons flowing out anteriorly to perform the action. Where are these coming from? This right here. But now, you're going to be like, what the hell is in the middle? You know it. Neural tube again, right? This guy. Eventually, it's going to be the cavity where cerebral spinal fluid is going to be stored. So this is where the CSF is going to be stored, right in the middle, right? And you know what the cerebral spinal fluid is for. It's for the buoyancy for the brain and the spinal cord. Very important. Now, how do we form the nerves and neurons going into this region? Because I said this is sensory, this is motor. Okay. Well, we need to figure out what is what. Okay, we said this is dorsal, right? Dorsal is posterior back. So the neural crest cells, here we go again. Neural crest cells, right? They're going to eventually form a dorsal root ganglia. That dorsal root ganglia is eventually going to form dorsal nerves. What are dorsal nerves? Nerves that flow into the dorsal side, so into the posterior gray horn. And what are those nerves? Well, they're right here. They're the peripheral nervous system. ANS and SNS, the parasympathetic and sympathetic nervous system. Neural crest cells. Okay, so, cool, I got that. How do you form the motor ones now? Not too bad either. So the floor ventral, bottom anterior. The ventral horn is then going to form ventral roots. Why? Well, if you have an innervation keep coming to the alar side, right, the posterior side, roots are going to start building from the bottom side, from the anterior gray horn, because they need to feed it somewhere. So these ventral roots are then going to form ventral nerves. These ventral nerves are going to be coming out, and they're going to start performing a function. What's their function? They use the neurons to perform muscle movement. Posterior gray horn, alar plate, sensory. Anterior gray horn, basal plate, right? Ventral roots, motor. Not bad. Not bad at all. I know it's really fast, but that's the best way I could explain it. Now, why did I do the bottom spinal cord first? Unfortunately, this is where worlds collide. What worlds collide? Brainstem and spinal cord, they need to collide, right? They're one in, what is that saying called? They're together, right? They're one in the same shell? I don't know. Okay, so their worlds collide, the brain and spinal cord interact with each other. How? All right. Remember I talked about the diagram. We formed the whole entire, the neural tube formed the proencephalon. Let me write them up here. Proencephalon. And then we formed the mesencephalon. Mesencephalon. And then we formed the rhombencephalon, right? Okay, and then the bottom region right here was the spinal cord. But now, since they're all part of the same neural tube, they gotta connect. So the proencephalon does telencephalon, diencephalon, we said it. Mesencephalon, chilling. Rhombencephalon does metencephalon and myelencephalon. I know these words repeat themselves, but I didn't make them. Okay, so the telencephalon, top one, right? He's gonna form the cerebral hemispheres. What are these guys? Cerebral hemispheres, going to be the lobes of our brain. We're going to form the frontal lobe, parietal lobe, temporal lobe, and occipital lobe. Where were these things? Well, frontal lobe was in the front, occipital lobe was in the back, parietal lobe was up at top, and temporal lobe was on the sides. Why am I saying sides? And why do I have times two? Well, the body's symmetrical. Unfortunately, it's not one lobe in the front of the brain, it's two. So we have two frontal lobes, two occipital lobes, two parietal lobes, and two temporal lobes. So it's really forming 8, but they just say it forms the 4 lobes, 4 main lobes of the brain. So telencephalon, cerebral hemispheres, lobes of the brain. Boom. Diencephalon forms the thalmi. What are thalmi? Thalamus, hypothalamus, posterior pituitary, and the optic vesicle. Okay, fine. We're descending now. So we have the top, now we have the thalamus, hypothalamus, optic vesicle by the eyes, right? So we're proceeding down. What's going to be next? Well, we're going to form the mesencephalon. It's going to be the midbrain, a little bit lower. This is where things get a little, I said worlds collide, here it is. See this structure right here? As your little diamond? Well, if you see the color, you notice we got red up top and purple on the bottom. What is that? Well, alar plate and basal plate, I had it red and purple. So I just took this structure, right over here, and I made it like a diamond, so it's a little simplistic. But just remember, this is the alar plates up top and the basal plates on the bottom. Okay. Unfortunately, the midbrain goes through this alar plate development, right? So the alar plate forms neuroblasts. These neuroblasts are going to form sensory neurons. Why sensory neurons? This is posterior. We know this is the posterior gray horn on top, right? Posterior gray horn was located up top. So boom, the alar plate is going to form the sensory neurons, like we said down here. These sensory neurons from the midbrain are going to form the superior and inferior colliculi. Okay, so you have a superior and inferior colliculi. Boom. Is the midbrain done? Unfortunately, no. Basal plate's going to form stuff too. How? Good catch. Neuroblast is going to form something. Well, we know everything's coming from the neuroblast. He's going to form the 3rd and 4th cranial nerves. Okay? He's going to also form the red nucleus. Maybe. They say maybe. I don't know. Substantia nigra and the reticular formation. Okay, so this is going to be the motor function from the basal plate. From the midbrain. Not bad. I mean, it's a little hectic, but what can we do? Now, rhombencephalon, right? Last one of the 5, or of the 3. The prosencephalon, mesencephalon, rhombencephalon. Divides again to the metencephalon and myelencephalon. Metencephalon, I am sorry, but he divides into 2 different structures. He divides into the pons and the cerebellum. Okay. Even though it's 2 different structures, it's not too bad. I'll show you why. The pons is the anterior part of the metencephalon. Makes sense? Metencephalon, anterior part, top part, right? And then cerebellum, posterior part, like back part. Okay, so top and bottom, not bad. So the anterior part of the metencephalon is going to form the alar plate. Well, also it's going to be built from the alar plate of the myelencephalon. Whoa, slow down. Metencephalon, right? Pons. Anterior part of it. Makes sense? But unfortunately, it takes the alar plate of the myelencephalon, so the bottom region right here. Why? I don't know. And what is that? Okay, well the alar plate we know is right up here. This is just another diamond, another alar plate and basal plate. But this time, this is from the myelencephalon. This was from the mesencephalon, the midbrain. This is from the myelencephalon. The rhombencephalon is where it comes from. Okay, what is he going to form? Well, the alar plate is going to form the sensory nuclei, obviously sensory, posterior gray horn. Sensory nuclei of the fifth vestibular and cochlear nuclei of the eighth cranial nerve. Okay, he's also going to form the pontine nucleus. Okay, so just remember the sensory region of the fifth vestibular and cochlear nuclei of the eighth nerves. Okay, the basal plate is going to form the motor nuclei of the fifth, sixth, and seventh cranial nerves. Why the motor nuclei? Basal plate, motor. Alar plate, sensory. Okay, so the pons forms the metencephalon, right? And it comes from the anterior part of the metencephalon. Sensory nuclei is the alar plate. Motor nuclei is the basal plate. The sensory stuff is the fifth vestibular and the cochlear nuclei and the eighth nerve. And the pontine nucleus. The basal plate, there's the motor nuclei of the fifth, sixth, and seventh cranial nerves. Whatever. Nothing else we could do, we have to deal with it. Cerebellum now, posterior part of the metencephalon. But also the alar plates of the metencephalon. Thankfully, we have our own guy. We have the metencephalon. The pons, I don't know why I had to use the myelencephalon, because he has to be extra. But the metencephalon uses the cerebellum, forms the cerebellum via the posterior part of the alar plates of the metencephalon, thankfully. So we're going to bring him right down here. Another diamond drawing. And something different about this guy? How are we going to form this? So the sides of the alar plates, they're going to bend, right? They're going to bend. The sides of the alar plates of the top region, the red, they're going to bend together medially, so together. And they're going to form the rhombic lips. Okay, so if you think rhombic lips, lips are together, so I guess the alar plates medially go together and they form the rhombic lips. Whatever. This is going to enlarge and unite in the middle, and what's he going to form? Well, he's going to form the cerebellum. How? The alar plates, right? They medially go together. They form together. Boom, cerebellum. Now we have the cerebellum formed. Okay, what could you do? Last one. Home stretch, baby. Here we go. Myelencephalon forms the medulla oblongata. Not bad. Unfortunately, got another diamond, so you know it's alar, and what's on the bottom? Basal. That's fine, not too bad. We know what alar is. We know alar is sensory. We know basal is motor. Posterior gray horn, anterior gray horn. We know this. Okay, so the neurons of the alar plate form the sensory nuclei, right? Obviously, from what cranial nerves? So they form the sensory nuclei for cranial nerve 5, 8, 9, and 10. Okay? It also forms the gracial and the cuneate nucleus. Also, the olivary nucleus arises from the alar plate. Something to memorize. So we have to memorize the gracial, the cuneate nucleus, olivary nucleus. All from the alar plate of the medulla oblongata. But the main thing to memorize. Sensory input for cranial nerves 5, 8, 9, and 10. I'm circling him. 5, 8, 9, and 10. Sensory. Alar plate, sensory. Boom. Now, last one. The basal plate for the medulla oblongata. He's going to form what? The neurons of the basal plate are going to form, obviously, 3 motor nuclei. So, obviously the motor nuclei of the cranial nerves 9, 10, 11, and 12 near the floor of the 4th ventricle. Why near the floor of the 4th ventricle? What did we talk about in cerebral spinal fluid? Okay, so you have 4 different ventricles, right? 1, 2, 3, and 4. Now the 4th ventricle is on the bottom, obviously. So the bottom of our brainstem is going to be where the 4th ventricle is going to be located. Same exact region. The top part, the prosencephalon, right, is going to be the 1st and 2nd ventricles. Lateral ventricles, you know it's left and right. Mesencephalon is going to be what? The 3rd ventricle, where the cerebral aqueduct is going to be. Okay, so this is all flowing. This is all in the same thing. Boom, up here, we're going to have lateral and right ventricles. This is for the cerebral spinal fluid, right? 1 and 2. Okay. The middle region is going to form the what? Well, this guy right here, he's going to form the 3rd ventricle. And where does he pass through? cerebral aqueduct. Okay, where am I going with this and why? Well, just good thing to like integrate everything. The fourth ventricle, the bottom one, fourth ventricle right here is the last one. It's going to be right by the medulla oblongata, like it says, near the floor of the fourth ventricle. That's the bottom of the brainstem. It's where the ventricle stops making cerebrospinal fluid, passes out into the dorsal sinus, out into the bloodstream. That's literally everything. I don't know. Like, I hope it helped. Like, if there's anything else I could talk about, maybe just know the different regions, know how everything flows, and we'll be good. Okay, so now we'll just do a couple questions to like recap everything we spoke about. You can skip through it if you don't want it. Okay, so here we go. A group of researchers is studying regional specifications of the developing brain. For the study, a rat embryo is injected with a drug that inhibits the area of the brain responsible for development of the midbrain. Okay, which of the following structures is most likely affected by this drug? I was like screaming it before, but let's just talk about it real quick. We know neural tube, right? Formed this region right here. 1, 2, right? Okay, and then we had the top region, which was known as the prosencephalon. Middle was the mesencephalon, and bottom was the rhomencephalon. Okay, and then this deviated into two different regions, right? The telencephalon and the diencephalon. This deviated into two regions too, the metin and the myelencephalon. And this guy stayed as the mesin. Does this give us our answer? Still no. This guy was the what? He forms the forebrain. Right, both of them. This guy forms the midbrain. And they formed the hindbrain. Okay, so it's asking us is where does the midbrain come from? Well, we know it comes from the mesencephalon. So this is going to be our answer. Diencephalon? No, that comes from the proencephalon. Metencephalon? No, that comes from the rhomencephalon. Myelencephalon? No, that comes from the rhomencephalon too. Telencephalon? No, this comes from the proencephalon. So the answer's got to be B. Let's see. Yes, that is correct. Okay, now, a 32-year-old man presents to the clinic for a physical examination. His skin is very pale. His eyes are blue. Interesting. What else do we have? And his hair is very light blonde, almost white. He says his sun burns easily and tries to avoid direct sun since it hurts his eyes. Physical examination is otherwise normal. Well, we know the guy's got albinism, right? Just by those symptoms, you could tell he has albinism. Now, what cells like make you albino? Well, what cells make you dark? Melanocytes make you dark, right? They change your skin pigment to make you lighter, darker, etc. Help you tan, you know that, the favorite ones. However, if you do not have these melanocytes, or if they're not active, you're going to become albino. Where do these melanocytes come from? That's what it's asking us. What germ cell layer gives rise to the cells affected by the patient's condition? Well, we know the cells are melanocytes. Okay. Endoderm. Maybe. I don't know. All of these are maybes, right? So let's just take it from the top. We knew when we formed this trilaminar disc, right? It then turned into something that was like that. It had the neural tube right here. The neural crest was around it. You have the meso, endo, and ectoderm surrounding that. But we knew that the neural crest, or the neural tube, formed the CNS. And the neural crest cells formed the PNS and ANS. It also formed a lot of different structures we talked about. One of those structures were the melanocytes. Okay, so if the neural crest formed the melanocytes, what else did they form? They formed Schwann cells. They formed the adrenal medulla, the meninges, the pianoarachnoid meninges, remember that? They formed the odontoblast, the endocardial cushion. They also formed the parasympathetic ganglia of the GI tract, right? It formed all those different things. But how is this helping us? Well, if we know the neural crest cells form these cells, we need to know where the neural crest cells come from. So let's work backwards. We have melanocytes. Okay, what forms melanocytes? Okay, neural crest cells. what forms neural crest cells? Well, if we remember, the neuroectoderm did. So neuroectoderm eventually forms these things. So C is going to be our answer. Let's see if we're right. Boom. Albinism, that's what we said the disease was, characterized by these conditions. And derived from neural crest cells, like we said, which come from the neuroectoderm. Good. Now, let's see. Okay, how much space we got? An 8-month-old child is brought to the neurologist by her mother after she realizes that the child is not responding to any sensory stimuli. Boom, your ear should light up. We scream sensory. The neurologist discovers that this is due to the failure of development of the alar plate. Obviously, we said alar plate, right? I'm going to try to draw in the same color if I remember. I'm going to draw the triangle thing because that's the easiest way. Red was alar, we said, I'm pretty sure, and purple was basal. Okay, so if we have the alar, and we have basal, boom. Failure of development of the alar plate. Which of the following failed to develop? Okay, so what was the alar plate? Well, it had things coming in, right? Let me draw a nice structure of it. So with a nice drawing, looks like this. And then we also had like that, if you remember. Okay. And then the alar plate was the red, right? And the basal plate was the purple. Okay, this is the alar plate was the posterior gray horn. And what was the basal? Well, that's going to be the anterior gray horn. This is going to be motor. So this was motor supply. This was sensory supply. Okay. But is it asking that? Well, it's telling us sensory supply, so we know it's posterior gray horn. And we need to know what structure didn't develop, because if it's not responding to sensory stimuli. Well, what comes out of the posterior gray horn? Well, we know the nerves that run through it are what type of nerves? Well, they're dorsal. And what comes through the anterior gray horn? Well, those are ventral. So it's not ventral. We know it's got to be something with dorsal. Okay, so alpha neurons, no. Beta neurons, no. Crus cerebri, no. Okay, so it's between dorsal horn and dorsal root ganglion. What do we know about the dorsal root ganglion? Well, the dorsal root ganglion doesn't come from the anterior gray horn. It comes from the neural crest cells. The neural crest cells form that ganglion, and that ganglion then could go through the posterior gray horn. But what is it going to pass through? It's going to pass through the dorsal horn. The dorsal horn is the thing that's receiving all of the stimulus. So if you're not responding to sensory stimuli, you have a defect in the dorsal horn. Who cares if the roots are there or not? You can't receive them. So it doesn't matter. If you can't receive them, or if you can't know that you're receiving them, this is the only thing that could be the answer. See if we're right? Yes, we are. Why wasn't E correct, like we said? Well, it's derived from the neural crest cells. That's not a good enough explanation. The best explanation is what I said. The dorsal horn is the thing that receives all of the flow. I think there's one more, maybe? No, that's it. All right, guys. Well, I hope the video helped, and